Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam
Waalaikumsalam
warahmatullahi
warahmatullahi
wabarakatuh.
wabarakatuh. Selamat pagi Pak Anto, selamat pagi Pak Endang dan juga Bu Arina. Also, good morning Dr. Sumpei.
Morning.
We are going to start our program UGC today. So, Assalamualaikum warahmatullahi wabarakatuh. Good morning everyone.
Good morning.
Welcome to the Undip Global Classroom. My name is Arif Rizky Nafisa and today I will be the MC and also the moderator for the UGC program today. We are going to learn and discuss an interesting topic entitled Modern Microbiological Research and Unveiling the Potential of Traucocytrid. Before we begin our main program, first we would like to invite Dr. Renat Anto Budiarjo, SSC Biotech, to deliver the opening remarks. The time is yours. Thank you.
Okay, thank you very much, Bu Arifa. Thank you very much, Dr. Sumpe, Prof. Endang, and also Bu Rina maybe, and other lecturer, and also my student. Uh, I thank Dr. Sumpe for your time twice to give like Undip Global Classroom in the topic of industrial microbiology. Dr. Sumpe is my colleague from University of Malaysia, Terengganu. He has been here twice, right, Dr. Sumpe? 2024 and 2025. And right now, there are like four students from biology department, three bachelor degree, and one just arrived last today or last night? Yesterday. Okay, so yesterday just arrived to do research in Terengganu with the topic of Trautocitrid. So, Trautocitrid is like new microorganism that not new in the term of the finding, but for Indonesia researcher, not many scientists research on these Trautocitrid. And it's a pleasure for me to work with Dr. Sumpe for the Trautocitrid project. He has been working with Rasta City Project, and we've been also working so far. And I hope with this Undipul Bus Classroom, the student will know what is Trausto Citrus, and yeah, maybe one of you want to do a research about Trausto Citrus here in Indonesia or also in Terengganu. Please feel free to directly ask me or Dr. Sumpe if later you want to have your final project with this project with this topic. Okay, I think that's all for me, Bu Arifa. I give time back to you.
Thank you so much, Dr. Anto Budiarjo. And the next session, we will take a photo group session. Please, for all students to open the camera. Please open the camera before I take a photo session. Adik-adik tolong dinyalakan dulu untuk kameranya. Okay. We'll take a photo now. One, two, three. Okay, next. One, two, three. Okay. And the last, 1, 2, 3. Okay, thank you. Now we are moving to the main session of today's program. Before we begin the lecture, allow me to briefly introduce our speaker today. Now we have Dr. Sumpei Ihata. With us is a lecturer and researcher from the Faculty of Fisheries and Aquaculture Science, University Malaysia Terengganu, Malaysia. He earned in his PhD in bioresearch from MEE University Japan, and his research mainly focused on environmental microbiology, gut microbiome, and also the trout for aquaculture applications. Through his career, he has participated in the international research, also has a lot of publication. And today we are honored to have him here to share his knowledge and insights with us to the main topic, trout. Please welcome Dr. Sumpei Ihata.
Okay, thank you, Ibu Arifa and Pak Anto, and good morning, everyone. I'm the Shunpei from the UMT. So today and next week, I give to the talk about you all. So today I focus on the microbiome because my research is not only the transcriptid. I do the microbiome using the NGS. And another one is the transcriptase. So today's session, I will share you the microbiome. Then next week, I will share you about the transcriptase. Wait. Okay. So at first, briefly introduce myself. My name is Shunpei Iehata, Japanese, and I got a PhD more than 20 years ago. Sorry. 2005 to the 2010, I studied in Mie University in Japan, near the Nagoya or Osaka, Kyoto area. Then after got a PhD, I do postdoctoral researcher at Antofagasta University, Chile for 2 years in the South America. Then after that, 1 year I do the postdoc researcher at the University, Japan. Then 2014, so 12 years ago, I start a lecture at the UMT. So at first, microbiome research. So what is a microbiome? So microbiome is a community of microorganisms that live in a particular environment. So composed of the bacteria, virus, protozoa, fungi, archaea is one of the microorganisms. And their host is the human, animal, and the plant. Of course, the environmental sample like the water or sediment also their environment. But as the host is the human, animal, and plant. So what is the difference between microbiota and microbiome? Simply, microbiota is the living microorganism found in a defined environment such as the oral and the gut microbiota or the sediment or other environment. On the other hand, microbiome is the collection of genome from all microorganisms in the environment, which including not only the community of microorganisms, but also the microbial structure element, metabolite, and environmental condition. So simply nowadays the researcher still using the mix, like some researcher use the microbiota, but some researcher use the microbiome. But simply microbiota is just their cell. And the microbiome is normally used for NGS, next generation sequence. So, type of the microbiota or microbiome analysis is the culture-based method. This one is the traditional culture, just simply grow microorganisms on selected media to isolate and study individual study. And this application is the identification of specific microbes, studying the growth characteristic. But their limitation is only a small fraction of microbes can be cultured. So for the uh, s- microbiologists say only we can culture 0.1% of the whole microorganisms or the microbes or the bacteria because that we don't know the actual the environmental factor to grow the bacteria or microorganisms. And the other than the traditional culture spreading or the plating method, we use the enriched culture. This means the enhancing the growth of the specific group of microorganisms from the mixed community using the specialized media. And application is similar to the traditional one, isolation and study particular microbial group, but their limitation is maybe still miss many species because the enrichment is that we isolate a very specific target is a specific group of the microbes. Second one is a molecular technique. As you all know, the PCR. PCR is amplify the specific DNA sequence to detect the quantify the microbial presentation, microbes. And so PCR have some different types, like the quantitative PCR, qPCR. This one is a measure the quantity of the specific DNA or RNA sequence to quantify microbial population. So normal PCR, we just amplify the specific DNA. But the qPCR is we can estimate the abundance of the specific bacteria or virus or any other microbes population in the environment. Then number three is a sequencing based method is the nowadays the the nowadays we have the three generation. First generation is the Sanger sequence. Uh, this Sanger sequence is the traditional sequencing method for analyzing the individual DNA fragment. So normally the application is the sequence a specific gene or a small region of the genome to identify the isolated microorganisms. Even though not only the microorganisms, the still use for the fish or the identify the fish or any other organism also. But this one is a limitation is a very low throughput and more suited to the target analysis like the isolation or just identify simply identification. And next generation sequence is the high throughput sequencing the technology for comprehensive analysis of microbial communities. Inside the NGS, the also have the second generation sequence or the third generation sequencing. So NGS application is a 16S ribosomal RNA genes amplicon sequencing. This amplicon sequencing is identify and classify bacteria or archaea based on the 16S ribosomal RNA gene using the NGS. The other one is the metagenome sequencing. This metagenomic sequencing is a sequence all DNA in the environment. So including the bacteria, archaea, and the other microorganisms, even though the higher organism, animal DNA also, we analyze this, and not only the 16S or 18S, whole microbial or the environmental DNA. So how to say that? Output is a very different with the amplicon sequencing. Amplicon sequencing is just the using the 16S, but the metagenome is a whole DNA, not only the 16S. And similar to the metagenomic sequencing, metatranscriptome this one is a sequence all RNA, not DNA. So metatranscriptome can use to study gene expression and their functional activity in environment, or even though the single cell. But their limitation is require the complex bioinformatics for data analysis. Like we need sometimes we need a supercomputer, or we need to learn about coding. Then others than this is the metabolomics, is based on the metabolome. And the proteomics is based on the protein content. And the transcriptome and the fish is the fluorescence in situ hybridization. That one is we use the observe or the count bacteria or target microorganism using a fluorescence microscope. Okay, so about the variable region of the 16S ribosomal RNA gene. So 16S ribosomal RNA gene compose the 9 variable regions. And as you can see the figure, some researcher use the very specific area for bacteria or archaea. So normally we use a V3, V4 or V3 only or V3, V5 for identify the bacteria community or including the archaea. And if only using the V3, more specified to the bacteria only community. Okay. And next is the sample collection and the preservation for 16S ribosomal RNA gene analysis. At first, we collect a sample. So select the appropriate marine sample. If you want to know about the microbiome in the marine water, of course, you need to collect the marine water, not the sediment or others. But the marine water type is we need to filter to collect the microbiome inside the marine water. Then after that, we how to say that? After that, we proceed the DNA extraction or the further reaction. And even though the marine water, of course, the different depths, like the surface water and the bottom water, is that their microbiome construction is different. So if we want to know that what the difference, the depths.
Mohon ditunggu sebentar, sepertinya ada kendala jaringan dengan jaringannya Dr. Sumpai. Mohon maaf, Pak Anto, apakah bisa dibantu untuk menghubungi Dokter Sumpai kembali karena saat ini beliau sedang terlempar dari Zoom.
Ya, ini saya coba.
Baik, terima kasih, Pak.
We must do analyzing I will start with this one. Maybe you can carry remote. I don't want to do but let's start with this one. Tell me about the titles. Can you get some information from what? Sorry, I didn't know. I'll go out.
Oke, no problem, Dokter Sumpai.
So the I'm not sure until where you all listen because the in the my screen still the sharing the slide. So I want to know which part I talk. This slide I already talk or not?
Next, Dokter Sumpai.
This one
Yeah, this
or
one.
this one? Not yet.
Yeah,
Not okay,
not
okay.
yet.
So the so this slide, no, this presentation, I focus on the amplicon sequence. And maybe you all study already the variable region of the 16S ribosomal RNA gene. So ribosomal 16S ribosomal RNA gene compose a 9 variable region V1 to V9. Then we don't use mainly we use the V3 to V5 or V3 to V4 because this one is a more the variable region and many researchers use this region for the V3, V4 or V3, V5 for the archaea and the bacterial community. But if we really want to focus only bacteria, then normally we use the V3 region only. Okay. So next is the sample collection and the preservation for the NGS, especially the amplicon sequencing. At first, we collect a sample from the various marine sample or any environment sample. Example, if we want to know what kind of bacteria or archaea composition inside the marine water, we need to at first we filter the marine water. But of course, the surface and the middle or bottom water have the different microorganism composition. So if we want to know the water difference depend on the depth, then we need to correct the different depths, not only the surface. And if we want to compare the different location, like also we need to correct that individually. And for the NGS, we need to do the replicate, at least a 3 replicate. Best is a more than 5 replicate to analyze the further. And when we collect a sample, of course, we need to use the sterile materials because the bacteria and the bacteria, especially bacteria, is everywhere. So that means that easy to contaminate. And if the contamination, then we get the wrong result. And next is the preservation. Preservation also important because if we don't properly preserve, then the bacteria contamination or even though the bacteria themselves is still alive, so they are grow. And the different bacteria or archaea is a different growth speed. So that if the fast speed one, easy to grow and become the dominant. So we cannot, after that we collect the sample, we cannot keep the longer in the room temperature, even though the chiller, because some the bacteria is a cyclotrophic. So that means some bacteria still can grow in the chiller, low temperature. So normally we keep or the storage the store in the deep freezer, -80. So this one is very important for the further research, like the bacterial compound or something. And DNA case is just normally we just keep in the DNA sample themselves. But if we want to use the RNA, then we need to use the RNA data like this means the special region for the RNA stable. So if we want to use the RNA, we need to keep the special, we need to use the special region. The DNA just normally just keep in the deep freezer. So next is the DNA extraction and the sequencing technique. So nowadays, DNA extraction kit is very developed. But still the DNA extraction have the traditional way like the CTAP or phenol chloroform extraction. But as I say, the DNA extraction kit is a very developed and many company provide. So normally we use a DNA extraction kit because the traditional way takes the very long, sometimes the one whole day. But if we use a DNA extraction kit, maybe we can get a high quality DNA within the few hours. So many researchers use the DNA extraction kit. But of course, the DNA extraction kit is also have the limitation, like sometimes that we cannot recover all the DNA. So this one depends on the researcher. I prefer to use the DNA extraction kit for the water sample or some other sample. But if just the DNA extraction for the single bacteria or microorganism, I just use the traditional way. Then after DNA extraction, we do the PCR for amplicon sequencing and prepare the library. Then after library preparation, we provide to the next generation sequencer. Then after get the sequencing, finish the sequencing, we get the raw data. This raw data need to analyze, data analyze, and the assembly and the annotation to see the bacterial community or bacterial diversity or even though the predicted functional analysis. So this is the bioinformatics pipeline for 16S ribosomal RNA gene sequencing. So bioinformatics pipeline, we use the CHIME or MOTAR or CHIME 2, but nowadays the many researchers use the CHIME 2. So this is the bioinformatics compose the quality control and the filtering, and the alignment and the clustering and taxonomy assignment and data analysis, including the diversity analysis. So at first, I explained about the quality control and the filtering. NGS, not only the NGS sequencing, especially NGS, sorry, have the low quality leads also. So if we don't do the properly the quality check or filtering, including a low quality sequence, make the wrong result. So at first we check the quality control based on the mainly the quality and the potential error. That one is the chimeric sequencing and filter the low quality leads to remove. Then after quality check and the filtering, we we conduct the alignment and the clustering using the reference DNA database, such as the silver or green gene. And we construct the operational taxonomic unit, OTU, or artificial ASV, artificial sequencing unit, ASV. Then after construct the OTU or ASV with the align the reference name, then we construct a taxonomy to make the what kind of the bacteria archaea composed in our own sample. Then after that, of course, we need to do the data analysis and the interpretation using the including a statistical analysis to determine the alpha and the beta diversity. Alpha diversity is just we want to know what kind of the bacteria or archaea compose how many percentage or something. And beta diversity is that we compare different location or different samples, how different, how similar, different between among the different environment sample. And explore the microbial community composition and identify the factor influence the microbial diversity and the structure also after the finish the assignment of the assign the taxonomy. So alpha diversity is the richness, evenness, and diversity index. Richness is the total number of the different OTU or ASV present in a environment sample, and this indicates a variety of the microbial species. And evenness is the relative abundance of each OTU or ASV, indicating how evenly distributed the microbial species. And diversity index, like the Shannon or the Simpson, combine the richness and the evenness to provide a comprehensive measurement of the microbial diversity. And beta diversity analysis is comparing the microbial community composition. So to unveiling the community difference or the dissimilarity matrix on the visualize and interpret how the difference using the PCOA or NNMDS. And also we can understand the drivers that by comparing the microbial community across the different location or different sample, we can identify environmental factors like the salinity or pH or the nowadays the climate change or the how climate change influence the microbial community. Then factor influence the microbial community structure in my marine environment, including the nutrient availability and the physical factor like the temperature, salinity, water depth, and hydrodynamic process also influence the microbial community. And biotic interaction, the predation, competition, and the symbiotic relationship among the microbes as well as the large organisms can significantly shape microbial community structure. So, example, the spatial and the temporal the variation in marine microbial community is the spatial variation is the microorganisms community in the ocean vary across different habitats. Example, the microbial community in shallow coastal water differs significantly from the found in the deep sea water. And the temporal variation also same. Compare the monsoon season and the dry season, of course, are different. And even though the if want to check the phytoplankton bloom, of course, the nighttime, the sun, daytime is different. So if we want to, or you want to compare the temporal variation, you need to correct the daytime and the nighttime in the same point. Okay. Until now is just the introduce about what is the microbiome. Next is I want to share my own research. So far, I finished the microbiome research, the fish, this one is the carp, and the shrimp gut, and the polychaete, and the marine sponge, and of course, I do the water sample also. So at first, I want to share the characterization of the microbiome characterization with the polychaete gut and how environmental sample from the wetland in Terengganu. So why polychaete or marine worm? Marine worm has the important role in ecosystem, like the bioturbation or the biogeochemical cycle, like such as the nitrogen cycle. And this one is used for the recreational fish bait or aquaculture diet because they have the high quality of the pure polyunsaturated fatty acid. But less researcher, the research do the conduct the polychaete gut microbiome. And polychaete-derived microbes has an important role in bioremediation process, including the biosurfactant production and the PAH degradation and polycyclic aromatic hydrocarbon degradation. And still have the potential probiotics, some of the microbes inside the polychaete, and create a unique microbiome to enhance marine benthic biogeochemical process. So that means that possibly regulate or facilitate the biogeochemical cycle in marine benthic environment. So the polychaete, we use the Nipah worm because this one is a major benthos in the estuary and prey to other macrobenthic animals and the potential pollutant biomarker. And this Namaricastis in Terengganu is very unique. we cannot find this polychaete in the marine water or the sediment. We only can find this polychaete inside the rotten nipa, the coconut tree. So that means that maybe their gut microbiome is very unique. So in these studies, objective is to assess the microbiome of the nipa worm gut, inhabit the rotten nipa and nipa themselves in the water sample using the 16S ribosomal RNA gene amplicon sequence. And after that, we annotate the predicted metabolic function of the microbiome related to nipa worm gut and its environmental sample using the functional annotation of prokaryotic taxa, Paprotax database. So we collect a sample from the this wetland. Malaysia is this one, and Terengganu is the northern part of the northeast part of the Malaysia. Then city wetland is the another 1 hour from my university to there by drive. So at first sample collection, polychaete and the NIPA collect like this, and the seawater, environmental water, around 100 to 500 milliliter of the seawater filter. Then after that, this filter keep in the -80. Then after sample collection, we do the DNA extraction and 16S ribosomal RNA gene amplicon sequencing. Then after sequencing, we conduct the data analysis using a CHIME2, R, and the Paprotax and the microbiome analyst software. Then this is the result, biodiversity analysis. As you can see, the water sample is a bit far from the polychaete and the NIPA sample. But the NIPA and the polychaete sample is close each other. So we can see the taxonomic composition of the phylum level. You can see the water and NIPA compose the high bacteroid water. However, of course, the Proteobacteriota was identified as one of the predominant phylum in all of them. The dominant group in the polychaete gut was the Actinobacteriobiota and the Firmicutes and Proteobacteriota. On the other hand, the Bacteroidota and the Proteobacteriota were observed as the predominant phylum in NIPA and the water sample. More deep in, this one left cell is a linear discriminant analysis effect size analysis. This one is we can see the biomarker to high dimensional biomarker discovery and the explanation that identify the genomic feature based on the genus, characterizing the difference between two or more biological condition. Like this case is a different host. Then this study conduct to identify genus level core microbiome as a biomarker among the polychaete and the NIPA and the water. Then you can see the polychaete. Okay, two genus found from the polychaete, Rhodococcus and the Candida. Siphonematobacter. And four different genus such as the Useria, wait, Useria, sorry, Useria, this one, Useria and Demikinia is the one of the biomarker for the NIPA sample. and where the 7 different genus, including the Synechococcus or the Chloroplast or the SAR86, is the biomarker for water sample related to the microbiome. Okay. Then this is the predicted functional analysis based on the how to say that based on the amplicon sequence result. So for predicted functional gene, chemoheterotrophy and the aerobic chemoheterotrophy and the nitrate reduction. Sorry, nitrate reduction were composed as the majority predicted functional of all the sample. However, for the functional predictions, including the hydrocarbon degradation and the aliphatic non-methane hydrocarbon degradation and the aromatic compound and the aromatic hydrocarbon degradation in the polykit show the much higher than the other sample. At the minor functional group, this is a major functional group. It means the more than the 1% or the more than 5%. But this minor group, functional group, like the kishiranolytic or cellulolytic or sorry, kitchenolytic or cellulolytic or the kishinolytic activity show substantially higher relative abundance in the polycate and the NIPA sample. Okay. So conclusion is the predominant phylum in the polycate gut composed of the Actinobacteriota and the Firmicutes and the Protobacteria. And some unique or the higher in abundance genus composed in the polycate gut like the Rhodococcus, the Lombastia, or the E. coli, or the Candida siphonae matobacter is the unique or higher in abundance inside the gut, polycate gut. And some genus like the Rhizobiales and the Demikinia and the Saccharimonaderes is a share with NIPA. That means some of the bacteria derived from the NIPA. And the functional result is hydrocarbon degradation or the aromatic hydrocarbon degradation and the aromatic compound degradation and the aliphatic non-methane the hydrocarbon degradation and the hydrocarbon degradation is a high in polycate gut because of these kind of the bacteria produce this kind of the enzyme. Okay, and next is the impact of the environmental factor on microbial community associated with the marine sponges. So why the marine sponges? Because the marine sponges has an important member of the coral reef ecosystem, like the filtration of the seawater. They can, how much? They can filter 10 to 40,000 square meter per day. And DOM, dissolved organic matter recycle, or the provide the food and the habitat in the other fish or the sea turtle. So role the symbiotic microbes is the carbon or the sulfur or the phosphorus and the nitrogen related to the symbiotic microbes. And as you can see, the still low information about the sponge microbiome in Malaysia, not only Malaysia, ASEAN country. You can see here is the ASEAN country. but so far, very less research. The marine sponge research normally the focus on the Mediterranean or Caribbean seas or the Antarctica. So, in this study, we evaluate the microbial composition associated with the marine sponge under the at first different marine sponge because maybe the different sponge has a different bacteria or archaea composition and different sampling time and different location. So the different sampling time is a temporal and different location the spatial. So for correction for the marine sponge, we correct the Bidon Island or the Kara Island because we have the EMT station inside the island. Okay. And for the special one, we collect 3 different islands. Each island around 20 kilometer differences. Then sponge species, we use the 4 different sponge species. One is Aptos and the Neopetrosia or Theonera or Zestospongia. These 4 is the common marine sponge found in the Bidon Island or the Malaysia Terengganu area. Then sampling period for the temporal variation, we collect the dry season and pre-monsoon season and post-monsoon season because we cannot go to the island during the monsoon season. Then after we collect the marine sponge, we keep in the marine sponge inside the plastic bag with the environmental water. Then we bring back to the sample to the lab and conduct the DNA extraction. And same time, we collect the seawater also. So sample collection, after sample collection, DNA extraction, this case, we don't use the DNA extraction kit because we compare which one is a traditional and the DNA extraction kit, which one is a high recovery rate. Then CETA method, traditional method is a much higher abundance or the DNA concentration. So we use the DNA extraction using a traditional method. Then after that, this one is the same with the boric sample. So result, at first biodiversity analysis, different hosts. As you can see, the Theonera and the Neopetrosia and the Despospongia or the Aptos is that they show the different biodiversity. And of course, that totally different with the water sample. Then this is the taxonomic composition based on the phylum level. You can see the phylum marine sponge compose the flexus, chloroflexus. But water is a very rare, low. So previous reports is 20 to 40%, but our result is higher than the 40%, 50%. And next, the predominant is the proteobacteria, but a very less abundance, like the less than 20%. However, the water sample is the almost 50% is the proteobacteriota. And cyanobacteria also very low, but Aptos is higher. But in the water sample is of course the water have a lot of the cyanobacteria like the Synechococcus, so their abundance is very high. Then next is we do the predicted functional result analysis. Interestingly, predatory or exoparasitic function found high abundance in the marine sponge sample compared to the water sample. Water sample is only 0.3%. This one might be the Dero vibrio. This one is one of the, how to say that, predatory bacteria. And this one, you can see the high Dero vibrio abundance in all the marine sponge sample. That's why the predatory or exoparasitic function in the marine sponge show the high. Then next is the nitrification and the aerobic ammonia oxidation also high in marine sponge sample. This might be nitrosopumilus, one of the archaea. Abundance is higher in the marine sponge. And next is the phototrophy or the photoautotrophy or the oxygenic photoautotrophy, photosynthetic cyanobacteria also high, but are still higher in water sample. This might be cyanobacteria for the phototrophy or the Synechococcus sponge alarm group may be related to phototrophic functions. So conclusion for the different sponge species, predominant phylum is a Chloroflexus and the Proteobacteriota and the Cyanobacteria. And Vibrio contribute to the predatory or the exoparasitic function, and the Nitrospumilus contribute to the aerobic ammonia oxidation and nitrification. And the Synechococcus sponge alarm also contribute to the phototrophic function in marine sponge. And next, we do the seasonal variation, the temporal variation. In this time, we focus on the only two marine sponge, Aptos and the Zestospongia. So, as you can see, easy to see the Aptos and the Zestospongia is a totally different, and water sample also different. But you can see however, the seasonal plots scatter except Aptos in pre-monsoon season is a very diverse. It suggests that each marine sponge has a unique microbiome regardless of the seasonal differences. This one same thing can say the water sample. Then next is the taxonomy composition. This one, yeah. The predominant phylum in both marine sponge is the Chloroflexus through all seasons. You can see this one, more than 20%. And next is a Proteobacteria was predominant phylum in water sample, while it was the member of the core microbiome in marine sponge too. Then compared to the seawater sample, the relative abundance of the Proteobacteria in marine sponge is much lower. This suggests seasonal factors have a big impact on the abundance of the Proteobacteria. Then next is the Cyanobacteria. As well as the same with the Proteobacteria, Cyanobacteria also compose the core microbiome of the marine sponge, regardless the season. And Acidobacteriota was detected in only marine sponge sample, regardless of the season. This might be the host specificity of the Acidobacteria in marine sponge show the highest abundance in dry season. However, they exhibit a different behavior in pre and the post-monsoon season. That might be suggest actinobacteria may have the host specificity or the seasonal variation. Then next, okay, this is the yeah, okay. So conclusion of the seasonal variation is that they have the host specificity is the chloroflexes and the acidobacteria and the acidobacteriota and the proteobacteria, and unique genus in the bacteria spirocheta or the Dero bibliota found unique in the aptos and the Zestospongia, the polybacteria. And seasonal relation is a cyanobacteria. And the two marine sponges have the unique microbiome structure, and the abundance of the some taxa fluctuated by the seasonal factor, like the cyanobacteria. Then next is the different location sample. In this study, we focus only the Zestospongia. Then how, as well as the other one, the water sample and the marine sponge sample is a totally different the diversities. However, within the sample, their diversities are similar, even though the different location. Even though that you can see the water sample have the very different depend on the location. And this is the taxonomic composition. The perhentian and the capas and the cara this marine sponge compose the crane archaeota and Acidobacteriota and whereas which one? This one is the Chloroflexus and Proteobacteriota compose the core microbiome in the Zestospongia. A bit different between with the seasonal or the different marine sponge is the crane archaeota. Then this is a taxonomic composition of the phylum. So as you can see, the crane archaeota, Acidobacteriota, and the Chloroflexus is very much higher in the marine sponge. And conclusion of the different location is the predominant phylum is the Chloroflexus and the Protobacteria and the crane archaeota and Acidobacteriota. And unique microbiome is the Entotheonerota and the Gemma monadota and the Nitrospirota and the Pauci 34F and so on. Many the microbacteria or the archaea uniquely found from the Zestospongia. And location difference is that the perhentian sample, cyanobacteria abundance higher compared to the other sample. And the capas is the Acinobacteriota and the Gemma is higher in the capas. And color sample is a Pauci 34F is a higher abundance in the color compared to the other samples. Okay, thank you for your attention. Sorry, maybe a bit messy for my presentation. So if you have any question, I can answer. So thank you.
Thank you so much for the for the very insightful like hand or type your name in the Zoom chat box. And don't forget before asking your question, kindly mention your name and also your name. Okay, is there any question? If there is no question from student, might I ask a question for Dr. Sumpei maybe? Actually, I have a question about the sample store. Um, what is it? Where we have to store the sample in the freeze condition or in the minus degrees, or is it will be damage the sample or not?
Sorry. Deep freezer or the other than the deep freezer or the freezer?
In just in the freezer, just freezer, not deep freezer.
Oh, -20 also okay. The normal freezer is okay, but not so long time because if the you store freeze store and the thawing, if you do the many times, of course
Yeah.
the DNA is degraded.
Oh, the DNA will degrade if we in the freezer and in the
Yeah.
out of freezer.
Thawing, yeah. I mean if you keep the freezer, then if you do DNA extraction, then you keep back this one many times, freeze and thawing
Yeah.
is make the DNA degradation.
Okay.
So normally we keep the small particle, like the not only the 3 replicate, 5 replicate, or the if possible 10 replicate from the same sample location.
Oh,
Yeah,
so we
because
have to have the replication.
we're not sure we can get the DNA or not. So better
Hmm.
backup. We take the backup. So
Oh,
10 replicate, if 10 replicate, then 3 replicate or 4 replicate use. Then if okay, okay. But if no okay, mean the no DNA. Sometimes we cannot get the DNA. Then we need to use recover the backup sample. So
oh.
we once we use, then we cannot keep back to the freezer.
And how long we can store our sample in the freezer?
Uh, some researcher use ethanol,
Hmm?
and some my case is that I use the DNA and DNA storage water like the TE or something the buffer.
Oh, TE buffer.
TE
Okay.
buffer mixed with the TE buffer and the SDS or something.
Oh, we
But
can
that depend on the sample. If
depend on the sample.
filter sample, we just keep the filter themselves in the deep freezer. We don't put anything.
Oh, if we use a deep freezer, we don't put anything like buffer TE, just put the sample in the deep freezer.
Also okay. But
Also.
for the water sample, just
Oh.
filter only. But the marine sponge sample, we afraid degradation or something. Then we just put the TE buffer or the buffer, DNA preserve the buffer.
Hmm.
But if you have the RNA data or zymo preservation region, then you can keep this one and can keep in the room temperature. No need
Okay.
to keep in the deep freezer.
In the
But
deep freezer.
I don't have this kind of DNA, RNA stabilizing the region, so I keep the -80 deep freezer.
Okay. Okay, thank you so much, Dr. Sumpai, for your answer. And now for student, is there any question for Dr. Sumpai? Because it's very interesting topic today is about the research in microbiome or microbiota in marine. We know that Indonesia have a lot of marine, right?
Marine, not only marine, I went Undip and Pak Anto
Yeah.
bring me to where the island. I forget the name.
Kalimun Jawa,
Karimun Jawa,
maybe.
yeah.
Yeah.
Karimun Jawa. Then Karimun Jawa have a lot of the coral or marine sponge. So very
Oh.
attractive the research site. Even though the marine sponge, we can see the beach side. No need to dive in.
Oh, no need to diving. We just collect
Yeah,
the
that's
sample in the beach.
yeah. Also, of course, here, Malaysia, my area, we need to collect the marine sponge, need to dive in.
Yeah.
But Karimun Jawa, no need. So good for research site.
Yeah, because Karimun Jawa is so beautiful too.
Yeah, so nice.
Yeah. Okay. Is there any question for students? Chelsea? Okay.
Okay, thank you so much for this space. I would like to give you a question, sir. It's about maybe I would like to ask about what factors should be considered when maybe we choose a variable region for microbiome research.
Very region mean the different location.
In a different location.
That one is the, of course, the salinity, pH, and the temperature, and the nutritional the value. Of course, the different location is a different temperature, or the sometimes the different pH and the salinity not so different if the marine water. But if we want to compare the estuary and the fresh water or the marine water, of course, the salinity is one of the most influenced factor. And the other than the physical salinity, temperature, pH, nutrition value also important, like the total nitrogen or total phosphorus or total carbon abundance is the influence to the microbiome composition. Is okay, Chelsea?
Yeah. Okay.
Okay, sir. Thank you so much for the answer. Is it very, very clear?
Okay, thank you, Chelsea. Also, thank you, Dr. Sumpay, as the answering. Is there any question? Is there any other question for Dr. Sumpay? Okay, if there is no question again, so we have to move into the last sessions. So this is the last session for our UGC today, and we have UGC in the 19 May 2026 next weeks for the last UGC. Thank you for everyone for the active participant in discussion today. Yeah.
Before the finish, I want to advertise my own research because nowadays I do the how to say that water microbiome,
Hmm.
the fish gut microbiome, and even though
Yeah.
that we do not only the bacteria or archaea, we also do the 18S eukaryotic microbiome also. So
Oh,
that if any student interest to this kind of research, please contact Pak Anto to study under me. Like that. Nowadays, the three FIP final year students conduct their own research under me. So I'm glad to accept anyone who interested not only the transcriptory, only also the microbiome research. If any student interest, then please contact Pak Anto. Actually, we need a student manpower, not enough
okay.
student power.
Yeah. It's very
Yeah.
interesting information, Dr. Sumpay.
Yeah. And
Yeah.
next week, 19 May, I focus on the trust lead. Today is
Okay.
just I share the microbiome.
Okay. Okay, thank you, Dr. Sumpay. Ya, I will translate the Dr. Sumpay. Jadi, bagi mahasiswa yang menginginkan untuk melakukan riset, ini nanti bisa menghubungi Pak Anto, jadi nanti bisa berkolaborasi dengan Pak Anto dan Dr. Sumpay. Okay, so we move to the last session because now it's 12. Thank you everyone for joining the UGC sessions. Before we close the session, allow me to briefly summarize today's about the lecture in the UGC sessions. Today we learn about molecular microbiome analysis, especially the use of metagenomic and other to know the microbial communities in every samples. So this approach helps researchers understand the role of beneficial of the microbes in supporting maybe health, growth, and also sustainable aquaculture systems. Okay, we also learn about the potential toxicity in the next weeks, and this is the promising marine microorganism also. On behalf of the committee, I would like to sincerely thank you for the Dr. Sumpe Ihata as our speaker today, and also Dr. Renat Anto Budiharjo as a PIC of the UGC in the microbiology industry, and also for Professor Dr. Endang Kusdiantini and also Ibu Dr. Arina Trilunggani as a lecturer in the microbiology industry today. Okay, I'm Arifah Rizky Nafisa as MC and also the moderator today. Thank you and see you in the next weeks. Don't forget to join in the UGC class in microbiology industry in the next weeks with our insightful speaker, Dr. Sumpe Ihata. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you.
Thank you, Dr. Sumpe.
Thank you. Thank you so much.